So welcome to everyone to our monthly Asia Pacific Regional TB Journal Club. And it's great that we have, I think, a very exciting paper to be presented and a good representation from the region as well, which is fantastic given the ongoing burden of and challenges of TB in our region. So welcome to everyone. I'd like to start the meeting just by acknowledging the traditional owners on the lands on which we're all joining. For those of us where I am, that's the Wurundjeri people of the Kulin Nation, as well as the Bunurong people. Pay my respects to elders past, present and emerging, and acknowledge that the lands were never ceded, and also acknowledge the traditional owners of all the lands on which all of you are joining us. My name is Dr. Philip De Croo. I'm co-chairing today, and I'm joined by Dr. Gail Cross, who is my co-chair. I'll hand over to you, Gail, to take us through the next bit.
Great. Thank you, Phil. Thanks, everyone, for joining us today. I'm thrilled to have Glyzelle, as well as Dr. Maricel Glare here today to help us lead us through this paper, which I agree, Phil, is a very interesting paper with new treatments for drug-susceptible pulmonary TB. Glyzelle is a physician and medical researcher, so clinician researcher, and she works at the TBHIV Innovations and Clinical Research Foundation, which was founded by Dr. Glare. And this foundation works on TB, HIV, and other clinical studies in infectious diseases. They're a research organization. Dr. Glare, in particular, practices primary care, and she's committed to bringing clinical evidence into everyday healthcare. So it's with great pleasure, Glyzelle, I'll hand over to you, and I very much look forward to us dissecting this paper. Thank you.
Thank you so much, Dr. Gail and Philip. Good day, everyone. Thank you to ACTnet for the opportunity to present today's Regional TB Journal Club. Today, I'll be discussing a recently published paper from the Lancet Infectious Diseases entitled Efficacy and Safety of a 4-month Quabodepistat, Delamanid, and Bedaquiline Regimen for Drug-Susceptible Pulmonary Tuberculosis. The study was led by Rodney Dawson and colleagues and evaluates whether a novel regimen containing Quabodepistat can shorten treatment for drug-susceptible TB from the standard 6 months to 4 months while maintaining efficacy and safety. This topic is particularly relevant because treatment shortening remains one of the major priorities in TB drug development. While we've seen remarkable progress in drug-resistant TB, shortening therapy for drug-susceptible TB has proven much more difficult. Over the next hour, we'll review the rationale behind the study, critically appraise its methodology and results, discuss its strengths and limitations, and finally consider what these findings mean for future TB research and clinical practice. Before we examine the trial itself, let's first understand why shortening TB treatment remains an important unmet need. Tuberculosis remains one of the world's leading infectious causes of death despite the availability of highly effective treatment. The current standard regimen cures most patients, but 6 months of therapy is still lengthy. Long treatment duration contributes to poor adherence, treatment interruption, increased healthcare costs, and ultimately poorer patient outcomes. Over the past decade, multiple attempts to shorten drug-susceptible TB treatment to 4 months have failed because relapse rates increase despite encouraging early microbiological responses. Therefore, the challenge is no longer simply killing bacteria quickly. It is identifying new drugs that can produce a durable cure after a shorter treatment course. This study evaluates Quabodepistat, a first-in-class DPRE1 inhibitor, as part of a novel combination regimen designed specifically to address that challenge. So before discussing the trial, it's worth understanding what Quabodepistat is and why investigators believed it was a promising candidate. The investigational regimen evaluated in this study is referred to as DBQ. representing the combination of quabodepistat, delamanid, and bedaquiline. Quabodepistat is the new component of the regimen. It is a DPRE1 inhibitor with a novel mechanism targeting mycobacterial cell wall synthesis. Delamanid is a nitroimidazole with bactericidal activity, while bedaquiline inhibits mycobacterial ATP synthase and disrupts energy production. The rationale for combining these three drugs is that they act through distinct mechanisms, potentially providing strong antimycobacterial activity without relying on rifampicin or isoniazid. Throughout this presentation, when I refer to DBQ, I am referring to this three-drug investigational regimen. In this trial, it was evaluated as a 4-month treatment and compared with the standard 6-month RHEZ regimen. Of these three agents, quabodepistat is a novel intervention, so the next slide focuses on its mechanism and why it was selected for this regimen. Quabodepistat, again, is a first-in-line class inhibitor of DPRE1, an enzyme that plays an essential role in synthesizing arabinin, a major component of the mycobacterial cell wall. By blocking DPRE1, quabodepistat disrupts cell wall formation, ultimately weakening and killing Mycobacterium tuberculosis. Beyond its novel mechanism, quabodepistat attracted attention because earlier studies demonstrated two important characteristics. First, it showed promising early bactericidal activity, suggesting that it could rapidly reduce bacterial burden. Second, it demonstrated a favorable safety profile during early clinical development. These characteristics made it an attractive candidate for inclusion in treatment shortening regimens alongside delamanid and bedaquiline. That naturally raises the next question: what evidence supported advancing quabodepistat into this proof of concept trial? Before launching this phase 2b/c trial, investigators built a stepwise body of evidence supporting quabodepistat. Ascending dose studies in healthy volunteers showed that doses ranging from 3 to 480 milligrams were well tolerated. This was followed by a phase 1-2 study in patients with drug-susceptible TB, where quabodepistat monotherapy demonstrated high early bactericidal activity over 14 days. Interestingly, no clear dose-response relationship was observed. Preliminary exposure-response modeling subsequently identified 30 milligrams once daily as the optimal dose. Finally, combining quabodepistat with delamanid and bedaquiline for 14 days produced early bactericidal activity comparable to standard RHEZ while maintaining a favorable safety profile. Collectively, these findings provided the rationale to evaluate whether this regimen could successfully shorten treatment over an entire course of therapy. This was a multi-center, randomized, open-label, proof of concept, non-inferiority, phase 2b/c trial conducted across 6 clinical research sites in South Africa. Participants were randomized using a central interactive web response system with balanced block randomization in a 1:2:2:1 allocation ratio to receive one of 3 DBQ regimens or standard RHEZ therapy. Although treatment allocation was open label, microbiology laboratory personnel remained blinded to treatment assignment, helping reduce bias in the assessment of microbiological endpoints. An important feature of the study is that treatment duration differed between groups. The investigational DBQ regimens were administered for 4 months, whereas the control RHEZ regimen followed the conventional 6-month course. Follow-up continued until 12 months after randomization, allowing investigators to assess both microbiological response and longer-term clinical outcomes. With the study design established, the next step is to understand who was eligible to participate and how the study population was selected. Before interpreting the results, it's important to understand the population being studied. Eligible participants were adults between 18 and 65 years of age with newly diagnosed, bacteriologically confirmed, drug-susceptible pulmonary tuberculosis. Importantly, participants had no previous history of TB treatment, making this a treatment-naive population. Drug susceptibility to rifampicin and isoniazid was confirmed by sputum culture, and written informed consent was obtained from all participants. Several groups were excluded to ensure participant safety and reduce potential confounding. These included individuals with previous TB treatment, clinically significant comorbidities that could affect participation or safety, and pregnant or breastfeeding women. Overall, this was a relatively homogeneous population with uncomplicated drug-susceptible pulmonary TB, allowing the investigators to evaluate the efficacy of the investigational regimen under controlled conditions. Now that we've identified who was enrolled, the next question is what exactly the investigators were trying to measure. Every clinical trial begins with a clearly defined question, and the study endpoints are designed to answer that question. The primary objective was straightforward. It was to determine whether 4 months of DBQ could achieve efficacy and safety comparable to 6 months of standard RHEZ. To answer this, the investigators selected end of treatment sputum culture conversion as the primary endpoint. Specifically, participants needed to have culture conversion with no subsequent positive culture by the end of treatment. The study also evaluated several secondary microbiological endpoints, including week 8 culture conversion, development of drug resistance, and time to sputum culture conversion. Finally, the investigators included exploratory endpoints that look beyond microbiological response, namely favorable outcomes, relapse, and longer-term treatment success. These outcomes are clinically important, but as we'll discuss later, the study was not powered to formally compare them. Safety was assessed throughout the study using adverse events, laboratory tests, ECG findings, treatment discontinuation, and other standard clinical assessments over 12 months of follow-up. Having established what the investigators intended to measure, let's look at the treatments that participants actually received. Participants were randomized to one of 4 treatment groups. 3 investigational arms received quabodepestat at doses of 10, 30, and 90 milligrams once daily. Regardless of dose, all investigational groups also received the lamanid 100 milligrams once daily and bedaquiline, administered as 400 milligrams once daily for the first 2 weeks, followed by 200 milligrams 3 times weekly. The total duration for all DBQ regimens was 4 months. The control group received the standard RHEZ regimen for 6 months, consisting of the conventional intensive and continuation phases. Treatment was delivered under directly observed therapy or video observed therapy according to local practice, with adherence support maintained throughout treatment and follow-up. This design allowed the investigators to compare a shortened all-oral investigational regimen against the current standard of care. With the interventions established, the final methodological question is how the investigators determined whether the new regimen was successful. The study was designed as a proof of concept, non-inferiority trial meaning the goal was not to prove that DBQ was superior to standard therapy, but rather it was not unacceptably worse while offering the advantage of a shorter treatment duration. Because previous studies showed minimal differences among the three quabodepistod doses, the investigators pooled all DBQ groups for the primary comparison with RHEZ. This approach increased statistical power while avoiding multiple comparisons. The study assumed similar efficacy between DBQ and RHEZ and was powered at 84% to test non-inferiority using a 12% margin. Non-inferiority would be declared if the lower bound of the two-sided 80% confidence interval remained above negative 12%. The primary analysis was performed in the modified intention to treat population using the Mitinen-Nurminen method, stratified according to bilateral cavitation status. Safety outcomes, on the other hand, were summarized descriptively using frequencies and summary statistics. With the study methods complete, let's now turn to the results, beginning with participant enrollment and disposition throughout the trial. This consort diagram summarizes participant flow from screening through study completion. A total of 306 individuals were screened, of whom 184 were excluded, most commonly because of negative rifampicin susceptibility results, abnormal laboratory findings, or other eligibility criteria. Ultimately, 122 participants were randomized across the 4 treatment groups according to the planned allocation ratio. Treatment completion and follow-up rates were high across all groups, with only a few discontinuations and minimal loss to follow-up. Importantly, nearly all randomized participants were included in both the modified intention to treat and safety analysis, strengthening the reliability of the efficacy and safety findings that we will discuss next. Before examining efficacy outcomes, let's first confirm whether the treatment groups were comparable at baseline. Before interpreting treatment effects, we first need to ask whether the randomized groups were reasonably comparable at baseline. A total of 122 participants were randomized. Overall, this was a relatively young population, with a mean age of approximately 34 years and a mean body weight of about 56 kilograms. One clinically important feature is the extent of pulmonary disease. Nearly 3/4 of participants had cavitary disease on chest radiography, suggesting that this was not an exclusively mild or low-burden TB population. The proportion with cavitation varied somewhat across the groups, from 67% in the DBQ30 to 80% in the DBQ10 milligram group. But there was no major imbalance that clearly favored one treatment arm. Age and body weight were also broadly similar, although the 10 milligram group was slightly older on average. Given the relatively small group sizes, some numerical variation is expected. The main point is that the treatment groups were sufficiently comparable for the randomized efficacy comparison to be interpretable. However, because the control group only included 21 participants, randomization cannot completely eliminate the possibility of residual imbalance. With baseline comparability established, we can now examine the trial's primary efficacy result, which is the end of treatment sputum culture conversion. The primary endpoint was sputum culture conversion at the end of treatment in the modified intention to treat population. The most important comparison is not among the 3 individual quabodepistod doses, but between the pooled DBQ and standard RHEZ. In the pooled DBQ group, 96 of 100% evaluable participants achieved culture conversion, giving a response rate of 96%. In the RHEZ group, all 21 participants achieved culture conversion. This produced a risk difference of -4 percentage points, with an 80% confidence interval from -7.4 to +3.4 percentage points. Because the lower confidence limit remained above the pre-specified non-inferiority boundary of -12%, the study formally met its primary non-inferiority criterion. In practical terms, these results suggest that 4 months of DBQ retained strong microbiological activity compared with 6 months of standard treatment. However, the endpoint requires careful interpretation. Culture conversion at the end of treatment demonstrates microbiological response, but it does not, by itself, establish durable cure. A regimen can achieve excellent conversion and still have an unacceptable relapse rate after treatment ends. Therefore, this is an encouraging proof of concept signal, not yet definitive evidence that DBQ can replace the standard regimen in clinical practice. The pooled analysis answers the primary question, but the trial also evaluated whether increasing the carbodepistat improved efficacy. Looking across the individual dose groups, sputum culture conversion was high at all 3 carbodepistat doses. Conversion was 100% with 10 milligrams, 92.9% with 30 milligrams, and 97.4% with 90 milligrams. The Cochrane-Armitage trend test produced a p-value of 0.6, indicating no evidence of a dose-response relationship. We should avoid interpreting the 10 milligram dose as superior simply because it had a 100% response rate. That group only included 20 participants, so one additional unfavorable outcome would have materially changed the percentage. Similarly, the lower response observed in the 30 milligram group may reflect random variation rather than a true pharmacological difference. The more defensible interpretation is that efficacy appears to have plateaued across the doses studied. Increasing carbodepistat from 10 to 90 did not produce a clear improvement in end-of-treatment culture conversion. This has an important drug development implication. When efficacy is similar across doses, the preferred dose may ultimately be determined by safety, pharmacokinetics, tolerability, drug interactions, and formulation rather than efficacy alone. A lower effective dose could reduce drug exposure and potentially improve safety or manufacturing feasibility, but this trial was not large enough to make a definitive dose selection decision independently. Beyond the primary endpoint, the investigators also examined early culture conversion and whether resistance emerged during treatment. The investigators also evaluated 2 secondary efficacy outcomes: sputum culture conversion at week 8 and the emergence of acquired drug resistance during treatment. Looking first at week 8 culture conversion, approximately half of participants in each treatment group had already achieved culture conversion by this time point. In the pooled DBQ group, 51% of participants were culture negative at week 8, compared with 52.4% in the RHEZ group. Across the individual DBQ dose groups, week 8 culture conversion ranged from 40% in the DBQ10 group, 50% in the DBQ90 group, to 57.1% in the DBQ30 group. Although there is some numerical variation, the confidence intervals overlap substantially, and there is no clear dose-response relationship evident at this early time point. The second secondary outcome was acquired drug resistance. Importantly, no participant in any treatment group developed acquired resistance during treatment, including resistance to quabodepistad, bedaquiline, or delamanid. This is an encouraging finding because preserving the activity of novel anti-tuberculosis agents is essential for future regimen development. However, it should be interpreted cautiously. With only 121 participants in the MITT population, the study was not large enough to reliably detect rare resistance events, and participants received intensive adherence support under controlled clinical trial conditions. Overall, these secondary outcomes complement the primary efficacy findings. They demonstrate early microbiological activity by week 8 and no observed emergence of acquired drug resistance, while also highlighting the need for larger studies to better characterize resistance risk in broader patient populations. Early microbiological activity is certainly encouraging, but more clinically important question is whether these early responses translated into durable treatment success after therapy was completed. Let's now examine the exploratory long-term outcomes, including favorable outcome and relapse. We've now seen that DBQ achieves the primary microbiological endpoint. The next question is whether that translated into durable clinical success after treatment ended. These analyses were exploratory and assessed outcomes at 12 months after randomization. Numerically, favorable outcomes were lower in the pooled DBQ group at 83%, compared with 95% in the RHEZ group. Likewise, relapse occurred in 12% of participants receiving DBQ versus 5% in standard therapy. At first glance, these numbers may seem concerning. However, it's important to remember that the study was not powered to compare relapse or long-term clinical outcomes. The control group consisted of only 21 participants, so a difference of just 1 or 2 events substantially changes the percentages. Therefore, these results should be interpreted as signals rather than definitive evidence of inferiority. To me, this slide highlights why microbiological endpoints and clinical endpoints are not interchangeable. A regimen may achieve excellent culture conversion, but still require confirmation that patients remain cured after treatment has ended. This is precisely why the authors, and I would agree, recommend that phase 3 trials use relapse-based clinical endpoints rather than relying solely on end-of-treatment culture conversion. While efficacy is only one side of the equation, any shortened regimen must also demonstrate an acceptable safety profile. One important point raised by the authors is that this is not the first attempt to shorten treatment for drug-susceptible TB. The best example is the phase 3 REMOX-TB trial. Although moxifloxacin-containing regimens achieved faster bacterial clearance, they ultimately failed because relapse rates were higher. In other words, early microbiological success did not translate into durable cure. The current DBQ study shows a similar lesson. While the regimen met its primary endpoint of culture conversion, the exploratory long-term outcomes were numerically less favorable than standard therapy. This suggests that the 3-drug backbone is active, but may still require optimization, perhaps by extending treatment duration or adding another companion drug. The authors also placed this study alongside other contemporary regimen shortening efforts, including Truncate-TB, Simplicity-TB, and Pan-TB-XBOS. However, these trials differ substantially in design, patient populations, and primary endpoints. As a result, direct comparisons should be avoided. Overall, I think the most important message from this discussion is that this trial demonstrates the potential of the DBQ backbone, not that the regimen has already achieved the optimal 4-month treatment strategy. Having reviewed the efficacy findings, let's now turn to the safety profile of the investigational regimen, which is equally important when considering treatment shortening. Overall, the safety analysis was performed in all 121 participants who received at least one dose of study medication, regardless of whether they completed treatment. Starting with grade 3 or higher treatment emergent adverse events, these occurred in 14% of participants in the pooled DBQ group compared with 5% in the RHEZ group. Although numerically higher with DBQ, severe adverse events remained relatively uncommon overall. Looking specifically at serious adverse events, these were also uncommon. They occurred in 5% of participants in the pooled DBQ group, while no serious adverse events were reported in the RHEZ group. Importantly, the investigators did not identify any pattern suggesting a new or unexpected safety concern. Treatment discontinuation because of adverse events was rare, occurring in only one participant in the pooled DBQ group and none in the RHEZ group. Likewise, no deaths were considered related to the study drugs. Another notable finding is that treatment-related adverse events were actually less frequent in the pooled DBQ group than in the RHEZ group, 17% versus 29% respectively. This suggests that although more than grade 3 events were observed with DBQ, investigators attributed fewer adverse events to the study drugs themselves compared with standard therapy. The investigators also monitored several predefined safety domains. There were no cases of clinically significant QTc prolongation, no major hepatic safety signal, and no deaths related to study drugs. A mild decline in estimated glomerular filtration rate was observed in some participants receiving DBQ, but this did not emerge as a clinically significant safety signal and was identified as an area for continued evaluation in future studies. Overall, the authors concluded that no major new safety concerns emerged. While this phase 2 trial was not large enough to detect uncommon toxicities, the overall safety profile was considered acceptable for a proof of concept study and supported continued evaluation of the DBQ regimen in phase 3. Having reviewed the efficacy and safety results, the next question is how the investigators themselves interpreted these findings. Let's now examine the author's conclusions and the implications they drew from this proof of concept trial. The investigators concluded that the 4-month DBQ regimen successfully met its predefined non-inferiority criterion for the primary microbiological endpoint. They also emphasized that the regimen was generally well tolerated, with no major cardiac, hepatic, or renal safety concerns identified during the study. Importantly, the authors did not claim that DBQ is ready to replace standard therapy. Instead, they appropriately describe these findings as proof of concept supporting progression to a definitive phase 3 trial. That distinction is important. This study provides evidence that the regimen is promising enough to justify further development, not evidence that it should immediately change clinical practice. Overall, I think the author's conclusions are appropriately balanced and consistent with the data presented. They acknowledge both the encouraging efficacy findings and the remaining uncertainty regarding long-term durability. Beyond the author's conclusions, it's also important for us as readers to critically evaluate what this study did particularly well. Several features strengthen the credibility of this trial. First, the study employed a randomized, multi-center design with a pre-specified non-inferiority framework, reducing selection bias and providing a rigorous proof-of-concept evaluation. Second, the primary endpoint was objective. Sputum culture conversion is laboratory-based and less susceptible to observer bias than many clinical endpoints. Third, trial execution was excellent. Treatment completion was high, loss to follow-up was minimal, and efficacy and safety were systematically assessed throughout 12 months of follow-up. Finally, evaluating 3 quabodepestat dose levels within the same study provided valuable information for future dose selection. The absence of a clear dose-response relationship simplifies subsequent regimen development. Collectively, these strengths make the trial a strong phase 2 study and provide a strong foundation for moving into phase 3. Of course, no study is without limitations, and these are equally important when interpreting the findings. Several limitations should be considered before applying these findings to clinical practice. The first, and probably the most important, is the choice of primary endpoint. End of treatment culture conversion is a useful surrogate marker, but it does not guarantee durable cure or protection against relapse. Second, this was a relatively small proof of concept trial with an especially small control group. As a result, the study had limited power to evaluate relapse, favorable outcomes, and uncommon adverse events. Third, because the trial was open label, certain clinical decisions, particularly components of unfavorable outcome assessment, may have been susceptible to bias, even though microbiology staff remained blinded. Another limitation is external validity. The study was conducted exclusively in South Africa and excluded several important populations, including children, pregnant women, older adults, and people living with HIV. Therefore, the findings cannot yet be generalized to these groups. Finally, although the combination regimen appears promising, the individual contribution of quabodepestat beyond the early bactericidal activity studies has not yet been fully established. Taken together, these limitations do not diminish the value of the study, but they remind us that this remains a proof of concept phase 2 trial. The ongoing phase 3 trial is designed to address many of these remaining questions before the regimen can be considered for widespread clinical implementation. Having reviewed both the strengths and limitations, I'd now like to offer a balanced, critical appraisal of what these findings mean for TB drug development moving forward. First, regarding internal validity. I believe the study successfully answered its primary research question. The randomized multi-center design, objective microbiological endpoint, and excellent follow-up provide confidence in the primary efficacy result. The open-label design is a limitation, but it is less likely to influence the laboratory-confirmed primary endpoint than subjective clinical outcomes. Second, in terms of statistical validity, the non-inferiority analysis was appropriately planned and successfully achieved. However, the relatively small sample size, particularly the control arm, means that long-term outcomes, such as relapse, remain imprecisely estimated. Third is clinical relevance. These findings are certainly relevant for adults with uncomplicated, drug-susceptible TB, similar to those enrolled in this study. However, the results should not yet be extrapolated to children, people living with HIV, pregnant women, or other populations that were not represented. Finally, regarding future implications, I think this study has achieved exactly what a phase 2 proof of concept trial should accomplish. It has generated sufficient evidence to justify a definitive phase 3 trial while clearly identifying the questions that still need to be answered. Overall, I would consider this a scientifically robust proof of concept study that advances the field while recognizing that it is not yet practice changing. With that appraisal in mind, I'd like to conclude by reflecting on what these findings mean for the future of TB research. I think this trial carries several important messages beyond its individual results. First, it reinforces that treatment shortening for drug-susceptible TB remains an achievable goal. Although previous 4-month regimens have struggled, this study demonstrates that newer drug combinations continue to move the field forward. Second, quabodepestat appears to be a promising backbone for future regimens. When combined with delamanid and bedaquiline, it demonstrated encouraging efficacy and an acceptable safety profile. And the absence of a dose-response relationship simplifies future dose selection. Third, the study provides a strong scientific rationale for a phase 3 trial. However, that trial should shift its focus from surrogate microbiological endpoints toward durable clinical outcomes, such as relapse-free cure. Finally, this work addresses a broader global need. A shorter all-oral regimen has the potential to improve adherence, reduce treatment burden, and simplify TB care if future studies confirm these findings. At the same time, several important questions remain, particularly regarding quabodepestat's individual contribution, its performance in more diverse population, and its eventual place within evolving TB treatment strategies. These remaining uncertainties naturally lead to several discussion points. To summarize today's journal club, I would leave you with 5 key messages. First, this study demonstrates that a 4-month, all-oral DBQ regimen is feasible, achieving non-inferiority to standard 6-month therapy for the primary microbiological endpoint. Second, quabodepestat appears to be a promising component of future shortened regimens, with encouraging efficacy, acceptable tolerability, and no clear need for higher dosing. Third, despite these encouraging findings, this remains a proof of concept study rather than a practice-changing trial. Important limitations, including the surrogate endpoint, small sample size, and limited generalizability, must be acknowledged. Fourth, the study addresses an important global priority. Developing shorter, safer, and more convenient TB regimens has the potential to improve adherence and ultimately patient outcomes. Finally, the next milestone is clear. A well-powered phase 3 trial with relapse-based endpoints and broader participant populations to determine whether these encouraging microbiological findings translate into durable cure. Overall, I believe this trial represents an important step forward in TB drug development. It does not yet redefine the standard of care, but it provides strong justification for continued evaluation of DBQ as a future treatment option. With that, I'd like to thank everyone for your attention and participation throughout today's journal club. I'd also like to acknowledge ACTnet and the TBHIV Innovations and Clinical Research Foundation for the opportunity to present this paper. I appreciate your time and I'd be happy to hear your thoughts on the study and its implications for future TB treatment research. Thank you very much.
Thank you so much, Glyzelle. That was a very thorough, detailed, and in-depth analysis of the paper. I really appreciate it. I don't know if we've got any comments from the audience. If we do, I would appreciate if you could put your hand up. I might begin with a couple of questions that I had myself, Glyzelle. One thing that I didn't quite understand in this paper was that they said that they excluded pretreated TB. But in one of the tables, in the demographic table, they did have TB treatment history, and about 21 patients, it looked like, or participants appeared to have had drug-susceptible TB in the past, or MDR TB. There was one as well. I just wondered if this is whether I've misunderstood this particular section in the table.
In the inclusion-exclusion criteria, the participant, so 300 plus participants were actually screened, and then a couple of them were found to be drug susceptible but have started treatment in the past. So they were excluded in the trial. Some participants were found to be multi-drug resistant. So they're also excluded to be enrolled in the trial.
Okay. Yeah, I'm not sure I quite understand this 21 that's in the demographic table, but I'm also curious as to the other eligibility criteria in this trial. Do you agree that in trials such as this, which are proof of concept, that we should be immediately excluding pregnant women, children from trial entry? What do you think about that? Do you think we need to have preliminary data before we include these key affected populations?
Yes, I think we have to gather preliminary data before enrolling this group of participants, as this study is still a phase 2 trial. We want the trial to be as controlled as possible, so we limit comorbidities or specific groups such as children or older adults, as well as pregnant women. So the study and the authors actually encourage this group to be included in a bigger trial, such as the phase 3 trials.
Yeah. One of the other things I was reflecting on as you were speaking was also this in TB trials. It seems to be the bar is set quite high because often we have to have preclinical data. Then we start off with obviously phase 1 to ensure that in healthy volunteers it's safe. Then we have early bactericidal activity trials to make sure that the drug works in the first 14 days with sputum culture conversion. Then we have trials such as this, which is phase 2b, but often small, so proof of concept. And then we move on to phase 3 trials before, we have potentially practice-changing trials. And the length of time that people have, the duration from drug discovery to when we have the drug introduced into use is exceptionally long, it feels, and we have to go through so many trial stages, which I suppose is the most robust way, but also feels quite inefficient in terms of getting critical drugs to people who need it. I wondered if you, Glyzelle, or anyone else had any thoughts on this.
Hi, Gail.
Hi.
I'm right. So yeah, it's indeed so like the goal for us clinicians would really want for drug regimens to filter through our patients already in a faster way rather than this traditional way of doing things, which is why I support the adaptive trials, which somehow shortens the process um, of mixing and matching several regimens and then selecting those that work better and moving them forward to phase 3 right away. So this should be the way to go. And there's another process that they're doing now, the modeling before. Instead of a phase 2, they do like modeling. So yeah, and move on to phase 3 if the model is positive.
For both Maricel and for Glyzelle, where do you think this regimen, the DBQ regimen, fits in our treatment? Let's say we do the phase 3 trial and we do have evidence that it is equivalent or non-inferior to RHEZ. where do you think it will fit in as an alternative? One of the things, I'll preempt this by saying, one of the things that captured me in this trial was the fact that we have less treatment emergent side effects from the DBQ regimen. And myself as a TB clinician, what I find quite hard is that actually standard TB drugs are hard to take. There's lots of side effects that people experience from it. That makes me a little bit hopeful that if this regimen turns out to be something that we do use for drug-susceptible TB, not only will we be able to shorten durations, but even provide patients with an alternative, particularly for those who are at risk of, the side effects that we usually see. So I'll tell you that that's my point of view, but what do you think?
The company has moved this forward to an MDR TB regimen, though they have not decided to use it but it's being used by other consortiums for a DSTB regimen. So yeah, it could be, since it's and the regimen is so the regimen has shown to have culture conversion at the end of treatment, although the durable cure is not so clear. The standard is still better with durable cure. So maybe adding on another drug or something can make it can make the durable cure better. But the company itself has, like, we're involved with the phase 3 for a DRTB regimen. So they moved the phase 3 now is a DRTB regimen, which I think is still uh, an area with unmet need, although there's already a 6-month regimen for MDR-TB, but there's an emerging resistance already for one of the components of the 6-month regimen, VPAL and BPAL-N. So this would be a good alternative for a 6-month regimen, which develop resistance to the VD.
Yeah, I noticed that when I looked up the phase 3 trial, they're randomizing fluoroquinolone resistant MDR-TB and fluoroquinolone susceptible MDR-TB in the Quantum trial, which is the phase 3 that you're speaking of. I think I will go back to what Glyzelle said at the beginning, though. I'm a big advocate for better drugs for drug-susceptible TB still, and I do believe that we need better drugs and shorter treatment regimens. So I do hope that those trials that are enrolling patients with drug-susceptible TB continue to look at this regimen amongst others. But let me hand over to Phil because I've spoken enough.
Yeah, maybe just a couple of comments. I thought, firstly, just an interesting point, and sorry, I've got some background noise, so hopefully you can still hear. One was the dosing of delaminid in this was daily instead of the normal twice daily, which has been modeled, but it's kind of reassuring that the 300 milligrams once daily seems to have done pretty well. I thought it was interesting that they chose to do daily delaminid, but not use the daily bedaquiline dosing that has been approved, but maybe they'll follow that up separately. Maybe just to follow up on the, where does this regimen fit? And I think it'll be an interesting one. I think I have concerns with this regimen being pursued for drug-susceptible TB, given the emerging bedaquiline resistance. I just wonder whether wide use of this regimen, given although it's very small numbers, the relapse rates, it just raises some alarm bells. Obviously, we need a phase 3 to see where it exactly lands. But I wonder whether these newer drugs should be kept a little bit more studied in MDR as they're being done. But I think that's a challenging one, because maybe getting better regimens up front, is what we really need for a lot of patients. And we know there's a lot of patients getting left behind still with standard treatment. I also thought, the fact that it's a phase 2B, but they were still doing different dosing for a drug that they've also used in other trials was interesting, and then not seeing much of a dose difference, although the numbers are small, was still interesting. And I just wondered whether we might have got more out of this trial rather than randomizing in the way they did to have a little bit more in the control arm to compare. But again, it's early proof of concept, and I think with early proof of concept, we always want more data. But yeah, those were my sort of few comments. I guess maybe opening it up to the floor to see if there are other people whose thoughts on where this regimen might fit, and maybe just one comment. I do know that Quabo-Depistat is being looked at for compassionate use coming online soon for bedaquiline resistance patients. So we'll get more data as a single drug perhaps from those sort of regimens. Sorry, Gail, I cut you off.
Oh, I was just going to say one word of caution would be that there is emerging evidence, however, particularly in terms of compassionate access for bedaquiline resistance, is that the mutation, the RV0678 mutation, also has cross resistance to DPRE1 inhibitors. There was one paper in 2023 that suggested that. It wasn't specifically for Quabo-Depistat, it was for another related molecule in the same drug class. So I think that's something that we need to pay close attention to, because obviously, Phil, your comment on, perhaps we should reserve these drugs for MDR-TB is sound. The risk is that with a lot of the new moieties that are coming to trial, there is unfortunately cross resistance with developing bedaquiline resistance because of the mechanism of action. And the way that I see things is often if we were able to get really good drug-susceptible TB treatments and reduce the duration, we may be able to stop, prevent the development of MDR-TB. But we need huge ecological studies in order to show that, I think. And that is really the challenge, is being able to run those very, very big studies to show that it reduces MDR-TB rates. So those eco studies would be very, very hard to fund and prove. I'm sorry, I talk a lot, but if there is anyone in the audience who has any comments, please feel free. We're nearly at time.
Yeah, I'd like to second to Philip. Some of us may be concerned of the relapse rate that appears higher in the TBHIV arm in this trial, and it deserves attention but should be interpreted cautiously. Relapse was an exploratory endpoint with very few events, and this study was not powered for formal comparisons. The authors appropriately recommend confirmation in phase 3 trials, and I guess everybody would approve to that as well. And I also kind of questioned while reading the article of why was pooling necessary and why would they have to do these arms when they wouldn't be compared in multiple times, like multiplicity. So I guess pooling was pre-specified. I mean, definitely pooling was pre-specified in the protocol, and earlier studies did not demonstrate a clear dose response relationship. So the primary objective was to basically just evaluate the DBQ regimen concept rather than compare doses. Pooling also increased statistical precision and power for the non-inferiority analysis, which was achieved in this trial. So.
All right. Well, we're at time, so we better let everyone go. Thank you very, very much, Glyzelle, for presenting the paper today. It was excellent. Thank you, Maricel, for agreeing to be part of the TB Journal Club. I would welcome you with open arms again next year, please, to present a paper again. Thank you, everyone, for attending today. Thank you to my co-host, Phil, and thank you to Nashita as well.